Alright, hello everybody, and welcome to today's lesson in biology. And as you can see here today, we'll be looking at the cell cycle. Now this is just going to be a very brief explanation, if you will, or rather a non-in-depth version of it. So let's get started. Now, the cell cycle can be divided into three phases. Now, in these three phases, the cell undergoes different processes within itself, and that's basically how the cell functions, as well as divides and all that. So essentially, the three phases, let's get a different color in here. Three phases: interphase, green, mitosis, let's get red, and cytokinesis. Interphase, mitosis, cytokinesis. These are the three phases of the cell cycle, essentially, and we'll be going a little bit more in depth on in each of them. So let's get right on with interphase. Now, essentially, interphase. Let me just do some jot notes here. This is where the cell spends most of its time. So essentially, this is where the cell does its work, So different cells have different functions. So this is where the interphase part of the cell cycle is where the cell will be fulfilling those functions. And it can actually be split into three further stages, actually. And these stages are G1, G2, and the S phase. So let's talk about the G1 phase, I guess. So G1. Now remember, this is all part of interphase still. So this is the period after cell division. Now, the stages of cell division, I'm going to cover later, they're mitosis and cytokinesis. Those are the stages of cell division. Now, G1 part of interphase is the period after that cell division occurs. So this is where the cell grows, DNA is transcribed, and protein is synthesized. DNA transcription, replication, we'll talk about that later on, as well as protein synthesis. But just remember that this is where DNA equals transcribed, and protein is synthesized. All right. Let's talk about the S phase after, because it's usually the one that comes after. This is where that's not a good D. This is where DNA is replicated. There we go. So DNA replication again. We'll talk about that later. I just know this is where the DNA is replicated. So after the DNA has been transcribed, essentially transcribing means like decoding or putting it into a manner where the cell can actually use the DNA. And with that transcribed DNA, it's replicated in the S phase. And going on to the final stage of interphase, this is the G2 phase. G2. This is where the cell prepares for cell division. So, cell and again, this is just getting ready for mitosis and cytokinesis, because this is where the cell will divide itself and basically fulfill another part of that cell cycle. But again, this is all in terms of what the cell does. Just know that in the G2 phase of interphase, the cell prepares for cell division. Now, speaking of cell division, let's get on to mitosis. Mitosis. Now, this is the second stage, right, of cell division. Or, sorry, not cell division, the cell cycle. So, draw a little quick diagram here. It goes, blue, interphase, then mitosis, and cytokinesis. I'm not going to write the whole thing, but you know what I mean. Cytokinesis. So, right now, this is the second phase of the cell cycle, but this is the first phase of cell division. Mitosis. So, again, when we said before, this is where the cell is going to replicate itself and split apart. Or essentially in mitosis it replicates itself, and in cytokinesis it splits apart. So, and speaking of mitokinesis, it actually, there's four stages. Four stages inside mitosis. Similar to how interphase had 3 phases. The G1, S, and G2. Mitosis has 4 stages. So let's talk about those 4 stages. So the first stage of mitosis is prophase. So let me draw you a little quick diagram of a cell. Or it's not gonna be detailed, but it's just gonna help us with this. So now that we're here, what you need to know about prophase is that in this phase, you're gonna have the chromosomes within the cell are gonna become visible. Now, normally, they're not all bunched together. Normally how you would see a chromosome, or what you would think a chromosome would look like, is like this, right? The classic X shape with in the middle. Well, in prophase, it's gonna become like that shape. So, in terms of the chromosome itself, just need to know that these two lines, they're called sister chromatids. Now, these are strands of DNA, and like I said before, in prophase, they become visible because they wind together. And become like this in this shape. And as well, the nuclear envelope within the actual cell, you just need to know that it breaks apart. Again, we'll talk more in depth later on, but just we need to know that for now, that the sister chromatids, the chromosomes themselves, coil together and become visible under a microscope, I should say, and the nuclear envelope breaks down. Now, the next part of mitosis is the metaphase. Metaphase. Let's go back to that drawing. What the cell looks like now is, it's going to look like, right now anyway, it looks like this. It has some DNA put as chromosomes just in the middle, or anywhere basically, but it's in there, in the nucleus. And what's going to happen in this is, in metaphase, is the sister chromatids, they're going to come to the middle of the cell, and they're going to align in the middle. So what's going to happen is they're going to get ready to be pulled apart. And they're going to be pulled apart because there's going to be these structures, these tubule like structures, and they're going to attach to sister chromatids and pull them away from each other. They're going to pull them apart. So there's going to be more, obviously, than just the one chromosome, but it's going to be pulled away like this and like this. And what you're going to have is the sister chromatids are just going to be by themselves, and they're going to go over here to the poles. Now that's kind of talking a bit about the second phase, or the next phase anyway, which is called the anaphase. Now, like we were talking about in the cell, in the anaphase, the sister chromatids are actually pulled apart. So in metaphase, they were just being prepared to be pulled apart, but in anaphase, they are pulled apart. And at this point, now that they are pulled apart, they aren't called sister chromatids anymore necessarily. They're just called chromosomes now. You're just gonna have chromosome here, chromosome there. So just remember that what actually happened here was you had one whole chromosome before. Let's draw it again. There's one whole chromosome of just coiled up DNA. And in anaphase, what happened was those tubule structures, which are called spindle fibers, they attach to the chromosome, to sister chromatids, and they pull them apart. Right? Pull them apart. So now it's half of the DNA that was actually there, which are moved to the poles. So they move to the poles. Right? So that's essentially what happens within this phase, and I was referring to them as sister chromatids before because they were separated, but they are separated, but now while they're at the poles, you can call them chromosomes again. Chromosomes. So during all the the previous phases. So in metaphase and prophase, the two of these are referred to as a sister chromatid, but now they're chromosomes again, or they're called chromosomes again. So next phase now, it's called telophase. This is the last stage of mitosis, and essentially what happens is let's draw our cell back in again. You have I shouldn't draw like that. So the chromosomes that move to the poles there we go. So chromosomes that move to the poles start to unwind. So remember how I said that the chromosome itself is made up of these spinning or these winded up strands of DNA. So they start to unwind again, so they become invisible. And remember how in prophase, the nuclear envelope surrounding them started to break down. Well, they start reappearing again around them. And this is all in part because this is cell division, right? You're taking one cell and you're making two. You had just the one. I say one, you just had the one chromosome before. Or, yeah, you had the one chromosome before. And then now you have two. And to complete cell division, you have to actually divide the cell, right? So the breaking down of the nuclear envelope, or not the breaking down, the development of the nuclear envelope is going to help with that in the next phase, is the last phase of the cell cycle. I shouldn't say last phase, but the next stage of the cell cycle, cytokinesis. And you know the rest. I'm not going to write the rest of it. So in cytokinesis, the nuclear membrane pinches off, right? So recall how we had the cell. The nuclear membrane is going to pinch off, and by the end of it, you're going to end up with two cells, right? Yep. So it would be like this before, and the nuclear membrane, as it develops around the chromosome, and after it did, it's going to pinch off, and you're going to get two cells. And the two new cells are made, and then they go back into interphase again. So they start doing their work. So if it was a skin cell, it would do the work that a skin cell would do. If it was a cell in your stomach, it would do the work that would be done there, which are obviously two different things, right? Alright, so that's a brief explanation of how the cell cycle works, and I hope you guys enjoyed. Stay tuned for our next set of lessons next week.